Okay, good evening everyone. So today we are going to start our lecture Still the Unit of Life. Okay, and myself is Amresh Kumar, and you are watching right now Education Media YouTube channel. Okay, and my qualifications are MS Geology, CSIR Air, All India rank 104, and qualified MPSET from Madhya Pradesh, also qualified CTET, Central Teacher Eligibility Test. So today we are going to try to complete the part 1 of the cell unit. Okay, we'll try to complete the part 1, the unit of life. Okay, so before that we have to know the fundamental, or we can say cell definitions. Okay, so we have to know the first cell definitions. Okay, so basically a cell is the fundamental structure and function unit of all living organism. It means all the plants and animals. Okay, all the plants and animals are composed of cell. Okay, so it means all the plants and animals are composed of cell. Single unit is called as cell. Okay, so next we'll discuss about cell theory. Okay, so basically cell theory is given by two scientists, we can say M.J. Sladen and Theodor Swan. Okay, so there is two scientists, M.J. Sladen and Theodor Swan, given the cell theory. Okay, so what is cell theory? Let's discuss about it. Cell theory basically tells about how one animal came from the, or we can say animals and plants are composed of cells and product of the cell. So basically cell theory tells about all the animals. Okay, all the animals, whether it is unicellular or multicellular. Okay, there are two types of animals are there, unicellular organism, or can say, and multicellular organisms are there. Okay, so all the animals and plants, okay, they are composed of cells. Okay, or we can say they are formed of cells. Okay, so this is all about cell theory, and when they have given the statement in 1839. Okay, 1838 to 1839. Okay, so Malthus Sladen in 1838 observed that plants are composed of cells, and Theodor Swan observed that this cell had a thin layer, outer layer, or we can call it plasma membrane later. Okay, so he also found that plant cells have a cell wall. Okay, so this is all about cell theory, and later cell theory is expanded. Okay, we can say expanded. Okay, so cell theory later expanded by Rudolf Virchow. Okay, Virchow, and explained that, or we can say cell theory later expanded as that cell divided new cells are formed from pre-existing cells. Okay, so it means all the new cells, okay, all the new cells that are formed, actually from where they came from, or we can say they arise from, okay, arise from pre-existing cells. Okay, so it means all the new cells came from pre-existing cells with the help of cell division, with help of cell divisions. Okay, so you know very well cell division having two types. There is two types of cell divisions are there. One mitosis and another one is meiosis. Okay, one mitosis and another one is meiosis. Okay, so these are the basically two types of cell division which causes the cell formations. Okay, so statement of cell theory is given: all living organisms are composed of cells and product of the cells. Okay, and the next statement of the cell theory, or we can cell expanded as, or we can say modified as, all cells arise from pre-existing cells. Okay, and the single statement, or we can say it is attest as omnis cellula e cellula. Okay, so this is all about cell theory. So let's discuss about an overview of the cell. Okay, so what are the materials that cells contains? Okay, what are the materials Okay, what are the materials, or we can say composition of cell. Okay, composition of cells. So composition of cell, it means we basically discuss here what are the things that compose cell. Okay, so all cells contain basically cytoplasm and ribosomes, and other also like membrane. Okay, so all the cell contains, okay, the fluid. Okay, we call it cytoplasm. Okay, we call it cytoplasm. Then after that, all the cells having a boundary. Okay, boundary, and boundary of cell is called as, you know very well, plasma membrane. Plasma membrane or cell membrane. Okay, cell membrane. Okay, some of the plants, or we can some of the cell, like plant cell having a cell wall. Okay, with extra membrane. So another one is ribosome, which is very important. Okay, another one is ribosome. So it is a very basically very important cell organelle regarding the point of examination. Okay, so why? Because ribosomes don't possess any kind of membrane, or we can call it non-membranous. Okay, non-membranous or non-membrane bound organelles found in cytoplasm, chloroplast, mitochondria, and on rough endoplasmic reticulum. So basically these are the location where ribosomes basically found. Okay, like chloroplast, ribosomes. Okay, ribosome present in chloroplast, ribosome present in cytoplasm in free form. Okay, and ribosome also present in mitochondria. Okay, chloroplast, mitochondria, and RER, rough endoplasmic reticulum. Okay, so these are the basic locations of the ribosomes. Okay, so let's discuss other cell organelles, or we can say based on the functions or shape of the cell, cell shape. Okay, cell shape. So we can call it cell shapes. Okay, shapes of the cell. So there are different types of cell shapes are there, like nerve cell, we can say it is elongated form. Okay, muscle cells having different structure, nerve cells having different structure. Okay, bone cells having a different structure, like bone cells is composed of calcium and phosphate. Okay, okay, muscle cells is formed, or we can say composed of muscle fibers. Okay, muscle fibers. Okay, cardiac muscles is, you know very well, it is composed of cardiac cells, or you can say cardiac muscles. Okay, cardiac tissue. Gland cell is special cells which secrete something, secretions are there in gland cells. Okay, blood cells having a WBC, RBC. Okay, different types of cells are having a different shapes, like biconcave cells are the blood cells. Okay, and reproductive cells which are having a different shapes, like sperm cells having a sperm cell having a flagella. Okay, and ovum. Okay, so these are the different types, or we can say different shapes of the cells. Okay, in case of plant cells, we have different type of tissues, like parenchyma tissue, collenchyma tissue, and sclerenchyma tissue. Okay, so basically parenchyma tissue is mostly abundantly, we can call it abundantly. Okay, mostly found. Okay, or mostly found in plant. Okay, so mostly in plant, basically parenchyma tissue is found. Then after that, collenchyma tissue is found also. And one of the most important regarding the point of exam is sclerenchyma, or we can call dead tissues. Okay, dead tissues. Okay, these are also found in plant cell in outer covering, like bark. Okay, so like it means sclerenchyma tissue present in bark of the tree, and collenchyma tissue present in basically like midrib of the leaf. Okay, midrib of the leaf. Okay, parenchyma tissue present in, or we can call it lamina of the leaf. Okay, lamina of the leaf, green color portion of the leaf, in which parenchyma tissue present. Okay, so these are the following shapes of the cells. Okay, parenchyma tissues having the oval shape, okay, like that one. Okay, having a intercellular space. Okay, intercellular space. But collenchyma having also a oval shape, but having a very less intercellular space. Okay, okay, so very less intercellular space present in collenchyma, but having a or we can call it parenchyma having a higher intercellular space. Got it? I hope you are getting my point. Then after that, sclerenchyma tissues having zero intercellular space or lack of intercellular space. They are compactly arranged. Okay, they are compactly arranged space, no intercellular space. Okay, no intercellular space. It means they are very hard. Due to the lack of intercellular space, they are very stiff. Okay, hard. Okay, so these are the following basics of the cell shape and structure. Okay, so shapes of the we can call it prokaryotic cells. Okay, in before that we have studied eukaryotic shape, but here some of the shapes of the prokaryotic cells are there. Like in prokaryotic cells, basically we have to know that prokaryotic cells having bacteria. Okay, bacteria are the sole members of the prokaryotic cells. Okay, I hope you are getting my point. So bacteria have four basic shapes. The first one is bacillus, second one is coccus, third one is vibrio, and fourth one is spirillum. Okay, so you can see like that rod like shapes like that one. Okay, okay, this one is bacillus, spherical coccus. Okay, coccus, and some of the examples are streptococcus. Okay, streptococcus. Okay, streptococcus is the example of the coccus one. Okay, and vibrio having a comma shaped, comma like that one. Okay, comma having that one like vibrio. Okay, like that one. Okay, these are the vibrio shape. Okay, and last one is spirillum having a flagella, that one. Okay, like those bacteria, that one having a flagella, or we can say like that one. So these are having a spirillum shape. So these are the basic shapes of the prokaryotic cells. Okay, so we are moving to the next slide. Okay, so what are the cell organelles present in prokaryotic cells? Let's discuss about cell organelles. Okay, cell organelles. So basically cell organelles having basically all the prokaryotic cell having a cell envelope, or we can call it cell membrane. Okay, cell membrane. Then after that mesosome, or we can call it chromatophores, nucleoid instead of nucleus, acting as nucleus. Okay, flagella for the movement. Okay, for the movement, as you can see here, this one is the flagella, this one is the membrane. Okay, this is the outer covering. Okay, envelope, or we can say outer covering of the bacterial cell. Pili and fimbria. Okay, this also is used for the attachment. Okay, pili, these are the pili, multiple pili as there in the covering, outer covering of the cell, and we call it we can basically call it, or we can say function as attachment. Okay, attachment to the surface. Okay, then after that, one of the most common cell organelle present in all these type of cells, or we can say all the living organisms having a ribosomes. Okay, so as you can see here, like that one dot red dots are represented here as ribosomes. Okay, so all the dot or we can say multiple ribosomes are present in prokaryotic cell. But most important regarding the ribosomes, that there is two types of ribosomes are there, or we can say subunits of ribosomes are there. Okay, so basically in prokaryotic cells are there 70S type of ribosome is present. Okay, very important. Okay, 70S is a unit, or we can say type. Okay, type of ribosome, 70S type of ribosome is present in prokaryotic cell. and other things, inclusion bodies, which is present basically inside the we can say inside the plasma or cytoplasm of the cell. Okay, so cytoplasm basically here written as I already told you that cytoplasm is a fluid of the cell. Okay, so fluid of the cell we call it cytoplasm. Okay, clear? I hope you are getting my point. Then our next little bit we have to discuss about cell envelope. Okay, which is very important regarding the exam, or we can say competitive exam. Okay, so basically all the prokaryotic cells having a different types of cell envelope composition. Okay, cell envelope, or we can say cell membrane. Okay, cell membrane. Okay, cell membrane. So basically cell membrane having different protection, or we can say protective layer, like three different layers are present in a cell envelope, or we can say cell membrane. The outer covering is glycocalyx. Okay, glycocalyx. This one is outer covering, glycocalyx, and the next one is cell wall. Okay, the next one is inner the glycocalyx, there is cell wall, and after that plasma membrane, the innermost wall. Okay, innermost wall, or we can say membrane, present in prokaryotic cells. Okay, so cell wall having a different composition compared to the plasma membrane. Okay, different bacteria is having a different composition of the cell wall. Okay, like for example, like some of the cell wall composition of having a peptidoglycans. Okay, later we will discuss about it, which of the bacteria having a peptidoglycans. Okay, so these are the following composition of the cell envelope. Okay, and the next so basically on the basis of cell envelope, there is two types of bacteria are there. Okay, bacteria classifications are there. Okay, and gram positive, the first one is gram positive and the second one is gram negative. Okay, so it is very easy to understand it. Like gram positive, they take up the retain the gram stain. Okay, and gram negative, they do not retain the gram stain. Okay, so stain is there. Okay, okay, for example there is a petri dish. Okay, and we have added stain. Okay, and the stain name basically violet stain. Okay, later we will discuss it. Okay, so we have added the stain, and later we have added the bacteria. Okay, suppose the bacteria got the stain. Okay, got the stain, it means it is positive one. If the bacteria did not stained, or we can say did not got any kind of stain, then it is mean if not got any stain. Okay, then we call it negative. Okay, negative stain. So this is all about gram positive and gram negative bacteria classification. Okay, on the basis of the staining process. Okay, like Escherichia coli is gram negative. It means they did not take the stain. Did not take the stain. Okay, but at the same time Staphylococcus are the positive, gram positive. Okay, gram positive, it means they retain the, okay, they retain the stain. They retain the stain. Okay, so this is all about little about we can say staining of the bacteria and classification of the bacteria on the basis of the stain, and it is proposed by the Christian Gram. Okay, Christian Gram. So next our next important cellular organelle is nucleate. Okay, which is nucleate. Okay, so as you aware that prokaryotic cells, what is the means of prokaryotic? Pro means earlier, early. Okay, primitive, or we can say primitive. Okay, karyotic, basically karyotic means, karyotic basically came from the karyon. Okay, karyon means nucleus. So basically prokaryotic cells having a prokaryotic cells have primitive nucleus. Okay, prokaryotic cells having a primitive nucleus, or we can say early formed nucleus. Okay, early formed nucleus. So the main character of the primitive nucleus that they did not possess, okay, possess nuclear membrane. Okay, it means nucleus do not have any kind of nuclear membrane. Okay, so genetic material basically spread, scattered like that one, okay, in the whole cytoplasm of the cell. Okay, so basically nucleoid having a genetic material, or we can say DNA. Okay, so it is formed of non-membranous, I already told you, or we can say I mentioned here like that one. Okay, nuclear membrane did not possess any kind of nuclear membrane, so we call it non-membranous. Okay, naked circular genomic DNA, single chromosome genetic material and protein. Okay, so this is nucleate having a, we say basically present in the center of the cell, and did not possess any kind of nuclear membrane. Okay, so it means all the genetic material basically is scattered in the center of the nucleus. Okay, center of the nucleus scattered. Okay, so, and attached with the help of proteins. Okay, DNA is attached with the help of proteins. Okay, so many bacterial have a small circular DNA, and we call it plasmid outside of the genomic DNA, and like that one. Okay, for example, like that there is presence of plasmid. Okay, so plasmid is also called as extracircular, okay, extra chromosomal, okay, extra chromosomal circular DNA. Okay, so what is the definition of plasmid? We can call it extra chromosomal circular DNA. So basically it is the definition of the plasmid, which is present in some bacteria, for example like E. coli, okay, and other, or we can say E. coli, the full form of the E. coli is Escherichia coli. Okay, so these Escherichia coli having a extra chromosomal circular DNA, and we call it plasmid. Okay, so these all about nucleoid, and the nucleoid having a we can say genomic DNA. Okay, so later we will discuss about it, DNA, what is the function of the DNA, and later. Okay, so next our next cellular organelles which is attached to the membrane, we call it flagella. Okay, we call it flagella. So basically in the previous slide I have already given the function of the flagella. It helps in, okay, it helps in movement, okay, movement of the cell. Okay, like if the cell wanted to move from one place to another place, they basically use the flagellar movement. Okay, so these so flagella are the thin filamentous extension from the cell wall of motile bacteria. Okay, motile means able to move. Okay, so it means there are few bacteria also, though they do not having a flagella, like E. coli. Okay, E. coli do not have any kind of flagella. Say they are not able to generally move like those bacteria having a flagella. Okay, so their number and arrangements are varied in different bacteria. Okay, like bacterial flagellum has 3 parts, which is very important regarding the exam point of view. Okay, so filament, hook, and basal body. Okay, this one is the filament, which basically move. Okay, so movement. Okay, and hook, one portion from where the flagella is attached to the basal body, and basal body is attached to the membrane. Okay, membrane of the bacteria. Okay, so this is the flagella. Multiple flagella can be present like that one. Okay, okay, so these are the filamentous extension of the bacterial cell. And another one which is very important for the movement and attachment, of the bacterial cells. Okay, so that one is pili. Okay, and smaller cilia like structure. Okay, so these are the elongated tubular structure made of special protein, and we call it pilin. Okay, anyway, so pili are like here like a structure. Okay, here like structure. Okay, like cilia one. Okay, like cilia one. So they are present in the outer covering. Okay, outer covering of the cell, as you can see here, like these are the pili, multiple. Okay, multiple, these are the pili. Okay, present in the outer covering. And fimbri. Okay, so basically fimbri and pili both are basically help in attachment. Okay, attachment of the cell, attachment of the cell to the surface. Okay, suppose any bacteria entered inside the host and wanted to reside inside a specific place, say, so bacteria basically try to attach. Okay, try to attach to the surface. So basically pili and fimbriae, okay, bristles, or we can say hair like structure, basically help in attachment of the bacteria to the any surface. Okay, I hope you're getting my point. Okay, so let's discuss a little about eukaryotic cells. Okay, so we have cleared the point of the prokaryotic cells. Okay, let me revise for you. Okay, prokaryotic cells. Okay, early cells, or we can say early cells. Okay, so prokaryotic cells having a only single chromosomes, having a nucleoid, having a cytoplasm. Okay, having a cell wall, plasma membrane. Okay, as outer covering, as well as having a ribosomes. Okay, the ribosome basically used for the protein synthesis. Okay, now it is time to tell, or we can say discuss about the eukaryotic cells. Okay, so U means, okay, here U represent as true. Okay, and karyotic, I hope you are getting my point. In previous slide I already told you that karyotic basically came from the karyon. Okay, and karyon means nucleus. Okay, so those cells having a true nucleus. Okay, those cells having true nucleus, or we can say modern nucleus. Okay, okay, true nuclear represented here like presence of nuclear membrane. Okay, so all the eukaryotic cells having a nuclear membrane present inside the nucleus. Okay, or we can say nucleus having a nuclear membrane in the center of the cell, or we can say. Okay, so eukaryotic cells are most advanced type of cells compared to the prokaryotic cell. In case of like in chromosome, there are number of chromosomes present, or we can say higher number of chromosomes present inside the eukaryotic cells. And thereafter that there is a size increase in the eukaryotic cells compared to the prokaryotic cells. Okay, and mostly eukaryotic cells have double membrane cellular organs, cellular organelles. Okay, compared to the prokaryotic cells. Okay, prokaryotic cells do not possess, okay, do not possess any double membrane cell organelles. Okay, double membrane cellular organelles. I hope you are getting my point. Okay, so it means in eukaryotic cells all the cellular organelles having a double membrane. Okay, what are the examples like? Okay, what are the examples of the double membrane? Like chloroplast. Okay, chloroplast, mitochondria. Okay, mitochondria and nucleus. Okay, all three like nucleus, chloroplast, mitochondria having a double membrane structure. Okay, So let's discuss about the cellular organelles present inside the eukaryotic cells. Okay, so eukaryotic cells having a membrane bound nucleus and organelles, as I already told you. And okay, and presence of membrane gives clear compartmentalization of the cytoplasm, as I already told you that these are the advanced form. Okay, advanced cell, more complex compared to the prokaryotic cells, but they are still single cell. Okay, as we already told you that cells, okay, we are calling it cells, not rather we calling it tissue or organ. Okay, we already know that there is a group of cells, we call it tissue, and a group of tissue is called as organ. Okay, so these eukaryotic cells having a membrane bound nucleus, or we can call it nuclear membrane, presence of nuclear membrane. And then after that, presence of membrane gives clear compartmentalization of the cytoplasm, and they have complex locomotory and cytoskeleton structure. Okay, later we will discuss about the cytoskeleton structure. Okay, like cilia. Okay, cytoskeleton. Okay, microtubules. Okay, so anyway, so and their genetic material is organized into the chromosomes. Okay, there are multiple number of chromosomes are there. Okay, there are multiple number of chromosomes are present. Okay, like in prokaryotic cells there are only single chromosome present inside the prokaryotic cell, but in eukaryotic cells basically there are multiple chromosomes are present. For example, like human cell. Okay, human cells having how many chromosomes? Like 23 pairs of chromosomes are there. Okay, it means we have a higher length of DNA. Okay, but in case of prokaryotic cells there is lower length of the DNA present. Okay, so examples of the eukaryotic cells like protista. Okay, protista members, fungi, or we can say fungal cells, fungi, plant cell and animal cell. So all the plant cell, animal cell, fungi and protista, all are the members of the eukaryotic cell. So what left? Only left is Monera. Okay, kingdom Monera, and kingdom Monera having bacteria. Okay, so bacteria are the members of the Monera, and these are the prokaryotic cells. Okay, these are the prokaryotic cells. Okay, so what are the different cellular organelles? Okay, cellular organelles present inside the eukaryotic cells. Okay, so the first one is cell membrane, cell wall, endomembrane system like endoplasmic reticulum, Golgi apparatus, lysosome, vacuoles. Okay, mitochondria, plastids, ribosomes, cytoskeleton, cilia and flagella, centrosome and centrioles, nucleus and microbodies. Okay, so these are the following cellular organelles present inside the eukaryotic cells. Okay, in part 2, we will discuss about eukaryotic cell. Okay, in part 2 we will discuss about eukaryotic cells. Okay, rest of you. Okay, thanks for watching the video. Thank you.